In this week's lab, you're going to be again donning the hat of a biochemist who is interested in characterizing an enzyme. Now remember, an enzyme is a type of protein, usually a protein, that catalyzes biochemical reactions. And in trying to understand an enzyme, there's all sorts of questions that one would ask. How fast do they work? How specific are they? And what are their optimal conditions? So let's review enzymes. Most enzymes are proteins. As such, their structure depends upon a series of weak interactions: ionic bonds, hydrogen bonds, hydrophobic interactions. And it's these interactions that cause that protein to fold in the proper way. And the proper folding of the protein is what gives it its proper function, the ability to bind a very specific substrate, catalyze the reaction, converting that substrate into a product. Now, because enzymes are dependent on all of these weak interactions, anything that disrupts those interactions will disrupt the structure of the protein and therefore the function of the protein. And as it turns out, because of this, proteins, enzymes included, have optimal pHs and optimal temperatures. And they're not always the same. So, for example, let's take a look at the left here. And what we're looking at is we measured the rate of a reaction catalyzed by two different enzymes. One, a human enzyme that evolved at 37 degrees Celsius, and another enzyme of a heat-tolerant bacteria that evolved at 77 degrees Celsius. So we've got two enzymes that evolved at different temperatures. And we measured the rate of the reaction under different temperatures. And so what you see is the human enzyme that evolved at 37 degrees has its highest rate of reaction at, not coincidentally, around 37 degrees. And that if you stray away from that optimal temperature, you begin to lose structure and therefore lose activity of that enzyme. Now look at this. The enzyme that evolved at 77 degrees Celsius, it doesn't even show activity until close to when the human enzyme already lost all of its activity, already became pretty much denatured. And that if we look, the highest rate of reaction of this enzyme here in red, not coincidentally, corresponds to the temperature at which it evolved, 77 degrees Celsius. If you stray away from that temperature, too low or too high, you lose activity because you lose the structure of the protein because it denatures. Now, the same holds true for pH. pH can affect those weak interactions that we've discussed. So if you look at two different enzymes that exist within the same organism, us, but in areas that are of different pHs, a stomach enzyme here and an intestinal enzyme, what you'll find is that it has its highest rate of reaction right there at the pH at which the stomach is at. pH 2 for the stomach enzyme and pH 8 for the intestinal enzyme. If you stray away from those optimal pHs, you are going to denature that protein, and therefore the protein will lose structure and function, and it will no longer be able to catalyze that reaction anymore. All right, now the enzyme that you guys are going to be working with today is phosphorylase that you are going to purify from potatoes. And what phosphorylase catalyzes is taking a starch primer, that's a couple glucose molecules put together, a molecule of glucose 1-phosphate, and it's going to turn that into a molecule of starch, and then an inorganic phosphate is sort of a byproduct of the reaction. So phosphorylase is the enzyme that we're going to ask our question about, and that question is, does phosphorylase have an optimal pH and an optimal temperature? And if so, what is the optimal pH and what is the optimal temperature? Now remember, anytime you are doing an experiment, you need to define your assay. What it is you are going to measure, what is the data that you are going to compare and analyze in order to see whether your experimental results matches, or your experimental outcome matches your predicted outcome. And for this experiment, we are measuring the endpoints using the iodine test. Okay, so let's talk about the iodine test first. The iodine test is using iodine to detect the presence of starch. Remember, starch being the product of our phosphorylase reaction. Now, an endpoint is a little bit trickier. Here's what the endpoint is. What we're going to do is we're going to add all of the reagents necessary to get this reaction to go. We're going to add our enzyme and starch primer. Now they are combined within the extract that you purified from potatoes. So along with the enzyme phosphorylase, you get starch primer. So you got those two already together. And then you add glucose 1-phosphate. And then immediately you take a little bit of that reaction and you add it to a spot plate. And then to that spot plate, that little bit of the reaction, you also add iodine. And then you see, did that iodine turn purple? Did it turn the reaction purple? If it did, awesome. That means starch has been made and we've detected it. If not, it doesn't turn purple. So that is the initial, right when we add the components together, immediately we take it out. That's our time point 0 minutes. And then we wait 2 minutes, do it again. Take a little bit of the reaction out, put it in the spot plate, take a drop of iodine, add it there. Does it turn purple? No? All right, let's keep going. Another 2 minutes, boom, do the same thing. Aha, but look at this. It's now starting to turn purple. It's a little bit light. If it's a legit positive result, then our next time point should be a little bit darker. So let's wait another 2 minutes, take a little bit of the reaction out, add a little bit of iodine, and yep, looky there. So let's go back then to where it just started to turn purple. The first time point at which we detected starch via the iodine test, that's called our endpoint. So the reaction here that we've seen under these particular conditions, our endpoint was 4 minutes. Now let's take a look. Here might be the results that you get, where we have several different conditions. We've got the reaction set up at 0 degrees or 10 degrees or 25 degrees Celsius. And then every 2 minutes, starting at 0, we took a little bit of the reaction out, put it into a spot plate, added iodine, and see if we get a positive result. So there's 3 different sort of classifications of endpoints we might get. Let's think about it. What does a high endpoint mean, right? 18 degrees. What does really, really low endpoints mean? Something like, 18 minutes. What does a really low endpoint mean? Like 2 minutes. Or what does it mean when we reach no endpoint at all? So think about this, because these are the sorts of results that you can expect to see, and you need to know what they mean. Okay. Now, the procedure that you will do, that you will go through, is that you purify phosphorylase from potatoes. You will then add iodine to your controls to ensure the reagents aren't contaminated. So you'll have a phosphorylase extract, right? Nothing else, just that. Add some iodine to it. Should that test be positive or negative? I'll let you think about that. Take glucose 1-phosphate, the reactant, alone. Add iodine to it. What should the test be? Positive? Negative? Again, I'll let you think about that. Starch alone. Add iodine to it. What would you expect? And then the phosphorylase extract plus the glucose 1-phosphate. Add some iodine to it. what would you expect to see? What should you see? These are controls. You should see something very specific that you should know right off the bat before even doing the experiment. If they don't work as expected, then you've got contamination problems. Once you've figured all this out, then we're going to set up the reactions under specific conditions, different pHs and different temperatures. You'll record the endpoint, or you'll record that the endpoint wasn't reached if you didn't get an endpoint in 20 minutes for each condition. And then from there, you will write up your lab assignment.